Okay, good evening. Good evening. Welcome, everyone. It's just turned 6:30 by my clock here, and good news is, at least in Newport News, the hail and rainstorm has just subsided. So we look like we're in for a good evening. Welcome to the 2021 Virginia Health Services Naturally Speaking series. I'm Rebecca Kleinhampel, Executive Director of the Virginia Living Museum. Before we get started, of course, with every Zoom meeting, we have a tiny bit of housekeeping. This presentation is being recorded. A link will be sent out, emailed on Monday. Questions may be asked by using the question box at the bottom of your screen during the presentation. You can upvote using your thumb icon if you like a question that's already there. And if you'd fill out the survey at the end so that we can understand your candid thoughts and what you like and would like to see in the future, we'd love to hear that as well. So tonight we're honored to present Dr. Jessica Labonte. She hails from the Great North, Quebec, Canada, where she received her bachelor's and master's from Laval University and her PhD, University of British Columbia. Postdoctoral research work was done with Bigelow Laboratory, and right now, currently, she's an assistant professor at Texas A&M in Galveston. So, Dr. Labonte, your research focuses on the role of viruses and hosts in aquatic environments. You were nominated by your peers to be part of this Ocean Discovery Lecture Series. 106 scientists nationally to partake this year, so congratulations for that. We are here in Southeast Virginia and beyond, thanks to Zoom, wanting to hear about your studies about microbes. I know you work with bacteria and viruses from our previous conversation, which of course are ubiquitous on our planet Earth. They live under extreme conditions where animals cannot be found, such as high salinity levels and geothermal hot springs. So Dr. Labonte, with that short introduction, I would like you to share with us a bit of your work and help us understand why some like it hot.
Thank you very much for the kind introduction. Hi, everyone. I'm going to use my laser pointer because it's easier than the arrow, I find. And yes, today I'm going to tell you a little bit about the microbes that live in hydrothermal systems, so microbes that really like hot living. And this is part of the Ocean Discovery Lecture. That is, so I'm going to tell you a little bit about ocean drilling, scientific ocean drilling at the same time. And here on this picture, we have the JOIDES Resolution, which is one of the vessels. It's the US funded vessel that is part of the scientific ocean drilling. And I spent 60 days on this vessel, and I'm going to tell you a little bit about what we did during our time there. So you're probably wondering why. So in my research and in my view, the whole world is microbe centric. But so I see microbes everywhere, and because that's what I study, even though we can't see them. But when we're thinking about the subsea floor, the subsurface, it's even more important because there's only microbes that can live there. So throughout the water column, you're gonna see fish and shrimp and all sorts of large animals. But as you go deeper and deeper, the oxygen is also going lower, and there's no animal that can sustain life without oxygen. And below the seafloor, usually within the first couple of centimeters, there's no more oxygen. So there's a lot of microbes, because microbes, they can breathe oxygen, but a lot of them can breathe other things. They can breathe sulfur, they can breathe nitrogen. So because in this environment there's no oxygen, they can thrive using all the other resources. And hydrothermal systems are one of the systems that are found in the deep sea floor. So you like in my lab, we also study sediment, but today I wanted to tell you about hydrothermal vents. So what are hydrothermal vents? So here, I'm just gonna close the image. So you've probably heard images of black smokers and white smokers, and how are these formed? So you have the seawater that will penetrate the oceanic crust and will react with the magma, the magmatic fluid, and be transformed, because the composition of the seawater and the composition of the magma are very different. One is cold, the other one is very hot, has a lot of metals, a lot of chemicals, and they react together and they come out through these vents. As black smokers is when there's iron sulfide, because that's a black compound. You also have diffuse vent, where there's not as much mixing with the magma, but there's still a lot of transformation. And here in these diffuse vents, there's actually a lot of life that is not microbial. There's a lot of shrimps and worms that thrive in these environments because they are very rich in the nutrients that are produced by the mixing of seawater and the magma, but they're not as acidic or hot, because you can see the magma is 1200 degrees Celsius, and when it comes out through the vent, 350 degrees Celsius. Not a lot of things can actually live and sustain these high temperatures. Actually, there's no living organism that can sustain these temperatures, and I'm going to tell you a little bit more later about the limits of life in terms of temperature. But here, maybe it's 30 degrees, we're 37 degrees. We know microbes love our body, so they also love these regions at the bottom of the seafloor. And why is it important that we study them? So we've heard of volcanoes. We see that they erupt on land in Hawaii and Iceland, but the majority of the active volcanoes are actually at the bottom of the seafloor. So, so they are great places to go study all these interactions, and they are at the seafloor, and hydrothermal vents is one of these. And the way we can study the deep sea, and including these volcanoes, is through the IODPP program, or the International Ocean Discovery Program. So I'm going to tell you a little bit about this program, since also this presentation is funded by IODPP as well. So scientific ocean drilling program, the program started in the late 50s, early 60s, and it was early on, it was called Project Moho. And that was to reach the boundary between the earth crust and the magma, and try to find to go as deep as possible in the center of the earth. And this was the first project, and even today it hasn't been fully achieved, because when you get too close to the center, it's so hot that we can't really drill that far. So it is still a project of interest, because we haven't fully achieved it. And then it became the deep sea drilling program from 68 to 83. And the first part was solely US funded, but after 73 and up until today, it is an international collaboration. Then it changed name again, became the Ocean Drilling Program from 85 to 2003. And in 2003, that's when microbiologists started being invited on the ships. And that was the Integrated Ocean Drilling Program. And now we're in the last phase of the International Ocean Discovery Program. So we have two versions of IODPP, the Integrated Ocean Drilling Program and the International Ocean Discovery Program. And now we're approaching 2023. We have actually as a collaboration, the scientists have written a new science program that is the plan. What do we want to do and achieve in the next 50 years of ocean drilling? And there are multiple platforms that are available for scientists who want to go and explore the seafloor. The JOIDES resolution is operated by the US, and that one is a riserless platform. It's pretty much a boat that, a vessel that can drill, a jack of all trade of vessels. It can go almost everywhere, but not in very, very deep places, or if it's too shallow as well. The Chikyu is operated by Japan, and that one is a riser platform, which allows it to go as deep, like very deep. They actually reach over 3 kilometers below the seafloor of coring it through sediment. And sometimes you need specific missions, mission-specific platforms. If it's a place that is unusual and we wouldn't be able to drill with the JOIDES resolution or the Chikyu, and those are operated by a consortium of European countries. During my postdoc, I actually worked on samples from Expedition 357 to Atlantis Massif. Atlantis Massif is very interesting because it's a big rocky mountain in the middle of the Atlantic. And it's so hard, the rock is so hard that none of these vessels was able to actually drill through it because you need some traction for the drill bit to be able to turn and drill. So they actually build robots that were sent down to the seafloor to collect the cores, and the robots would then bring them back up. If anyone is interested to participate, there's many opportunities with IODP, including sailing as a shipboard scientist or technician. I'm going to tell you about my experience as a shipboard scientist. You can serve on an IODP science advisory panel. I actually served for 3 years. My term was up last summer on the US Advisory Committee. You can also request your own sample to do your own research on samples that were collected using the JOIDES resolution. And if you know of any graduate students, there are plenty of opportunities for them as well. They can sail, there are fellowships, they can assist in post-cruise research, and there is more information available at usoceandiscovery.org. So I'm going to tell you a little bit about my experience as a shipboard scientist on Expedition 376, which was called Brothers Arc Flux. So here we have the science party that while we were on the ship, we actually had 30 scientists, and it's a wide range of scientists from microbiologists like me to petrologists. There's people looking at the structure, the physical properties. So we were looking at all facets of our samples that were the rock that we got from the cores. Our expedition was in 2018 from May 5th to July 5th. So it's when you go on one of these expeditions, you're gone for 60 days at sea. You're not coming back, and we were working around the clock. I was on the shift from noon to midnight. As a person who always goes to bed at 9 p.m., I got to go to bed at 1:30, 2 a.m. for 60 days. So I'm good for a very long time of going to bed late. And we left from Auckland, New Zealand, and there was about 24 hours transit before we got to our destination, which was Brothers Volcano. So Brothers Volcano is off the coast of New Zealand, and it is found in the Kermadec Arc. So a volcanic arc, what is it? So when you have two tectonic plates and one of them goes under the other one, the one that's under it will actually start melting when it reaches the mantle. And that's going to actually push some of the melting fluids up, creating a volcano that is at the base of the seafloor. And all of these dots here, most of them are actually volcanoes. So Brothers is one of hundreds of volcanoes that are found in the Kermadec Arc. So this is a picture of Brothers. That's actually, we can see its shape. There is the upper cone, the lower cone, and this is the caldera. So a caldera, the way it's formed, so you're gonna have your chamber, then the magma is going to erupt, creating a volcano. But at some point, when there's too many too many places that are ejecting magma, it's making the rock collapse, because you're sort of creating like faults that will make it collapse, which will form a caldera, and then magma can come up again through these rocks that have collapsed and have another volcano that will be formed. So that's how Brothers Volcano was made. There was originally one volcano, and then the whole ground collapsed, forming the caldera, and then the upper cone and the lower cone actually went afterwards. So this is how this is another view of Brothers where you have the magmatic conduits that you can see with the upper, this is the caldera and the upper and lower cones. And we had samples from all of the, so when we, that's the view from the aerial view of the volcano, which we have here. And we got samples from 5 major locations. We had in the northwest caldera, and in the west caldera, the upper cone. We tried to drill the lower cone, but it just wouldn't work, and it was towards the end of the expedition. Somehow it was just, it kept on breaking. We didn't have good recovery, so we didn't get much samples towards the end of the expedition. So there were multiple, so whenever there's an IODP expedition, there are like defined scientific objectives that need to be achieved. And in this case, there were 4 objectives. The first one was to try to understand the characteristic of the volcano, like is the magma chamber, that are the volatiles coming from just one pass, or is there a mix? So when it says brine, is there a mix of salt water and the magma? How is this volcano? Like, it was trying to understand how this volcano has come to be, basically. Look at the precious metals and metalloids and how they have their interactions with the rock and with the magma have actually shaped the rock that are present today and at Brothers Volcano. Look at the fluid rock interactions, because when you have seawater reacting with rock, it will change the composition of the rock. And what I'm going to tell you about today is, this is the most relevant for microbiologists like me, was to assess the diversity, extent, and metabolic pathways of the microbial life in this extreme metal-oxic and acidic volcanic environment. Or what kind of microbe is crazy enough to live in a place that is super hot, that has a lot of toxic metals, and is also super acidic? So why are we studying these microbes in the hydrothermal vents? There are actually reasons, that big questions that can be answered by studying hydrothermal vents. So it is hypothesized that actually life originated in hydrothermal vents. So right now, when we think of hydrothermal vents, we think of them as an extreme environment because it's so hot. But at the time when you're thinking of times of early Earth, the surface, there wasn't any ozone yet. So the surface was bombarded by radiation, and it was actually much harsher at the surface than at the bottom of the seafloor. So that was a place where there was an amazing supply of nutrients, especially hydrogen sulfide, which microbes love. There were also these little micro like pockets in the rock that made these ideal environment where microbes could thrive in these little compartments. And the supply of energy was just amazing. So all of these, and also when you recreate in the laboratory the conditions at hydrothermal vents, the biological molecules actually, they auto-make themselves. So that's a good way to hypothesize, like that's why we hypothesize life may have originated there, because these biological molecules, they actually can be formed without microbes making them. They can be made by themselves because of the conditions that are present in the hydrothermal vents. So because life originated in hydrothermal vents, we can look at the microbes that are inhabiting these environments and try to place them on the tree of life. So this is the most updated view of the tree of life. We have the eukaryotes here. We have the archaea, all of these, and we have the bacteria. And what's interesting with this is that everywhere where there is a red dot, that means we know these microbes exist, but we don't know, like don't have any culture for them. So all we know is because we sequenced their genetic material, we looked at their DNA, so we know they are in the environment, but we don't have a physical tube with the microbes in them. So you can see that there's still a lot of unknown that we can discover in every environment we're going. Another reason why we study hydrothermal systems is to study the limits of life. So temperature is is currently on Earth the biggest limit of life. There are microbes that live, they can grow at 0 degrees Celsius. That's when things freeze, all the way to 120 degrees Celsius. Beyond that, we haven't found any life beyond 122 degrees Celsius. But hydrothermal vents we can look at those microbes called hyperthermophile, the microbes that live at very high temperature, more than 80 degrees Celsius. Another limit that is found in hydrothermal vents is that they are very acidic. So the pH is very important. So you can have microbes that live from 1 to 14. At Brothers, it was very acidic, and salinity is also one. And if you remember, in my first objective of the expedition, they wanted to look if there was any sort of brine fluid. So brine means salty, and at Brothers, they found that there was actually more. The water was, the salinity was higher in the fluids in the hydrothermal vents than they were in the upper column. So we can look at halophiles, acidophiles. The pH was less than 2, so that's very acidic, and the temperature was up to 350 degrees. We stopped measuring, and I put this picture here. So this is Cornel Duron. He was the one of the 2 chief scientists. And at one point, the drilling people, they were like, oh, there's something wrong. Well, it turns out that the harsh condition of Brothers Volcano degraded the drill pipe on the ship. So we can see it got all rusted out, and it actually broke. It corroded so much that it broke the drill pipe, and we were no longer able to actually drill. So these are pretty harsh conditions. So question we can ask, how hot is too hot for Earth life, Earth-style life? So temperature, and this is a picture from another expedition. It was an expedition that specifically looked at the temperature in the Nankai Trough. So there was a really nice gradient in this environment from 30 to 130 degrees Celsius. So we know the current limit is about 122. So they were able to look at all the microbes, all of there. We didn't necessarily have at Brothers a gradient, but we know it can go all the way to 350. So the microbes, the deeper we go in the core, the hotter it's going to be. But also, we have to be mindful that they may not be in the fluid, but the rock can create these micro pockets that would create the perfect environment for life, even if the outside may be a little hotter. I'm showing this because while we were on the ship, we were always asked, so every day we would have a scientist meeting, and every Monday, every group, whether they're petrologists, microbiologists, we had to present results. But as microbiologists, we would just take the samples and then just store them, because most of the work we have to do back in the lab. So we started making these Microbe Monday infographics where we would choose a microbe that could potentially be living at Brothers and present it to the geologists that were on board. So this one is Methanopyrus candleri. So it produces methane and fire, because this is the bacteria that can live at the hottest known temperature, 122 degrees Celsius. That can, knowing that water boils at 100 degrees Celsius, we have to add pressure to make sure the water is not boiling to be able to reach this hot temperature. So every Monday, we would show our Microbe Monday slide presenting a new microbe. I told you pH. So pH, like when you're using your cleaning products, it's usually basic. Our body, pure water is 7. And the gastric acid lemon juice, pH 1 or 2, that was the pH level that was found at Brothers Volcano. So we could be studying acidophiles. And here we have an acidophile that is very well studied called Sulfolobus sulfataricus. And sulf, because its favorite food is actually sulfur. It is commonly found in volcanoes, hot springs, and hydrothermal vents. pH, it's an acidophile, pH 2-3. This one has a few viruses that are actually studied. So viruses, this is what I study in my lab. They are very important. It's hard to, like, you may not believe me when we're in a COVID pandemic era, but without viruses, we wouldn't be there, because they play huge roles in the environment by recycling nutrients and also controlling the microbial populations. So here we had this virus SSV1 that when it has a very interesting spindle shape and make florets when we look at it under the microscope. So we presented this infographics. And the last one, this one is just a funny one, because Thermos sulfidibacter. So it's also an extremophile and it eats sulfur, but it's Thermos sulfidibacter takai, and Dr. Ken Takai, it's named after him. But he was also one of the microbiologists on board the JOIDES Resolution during the expedition at Brothers Volcano. So it was just a little wink at Ken Takai, who was part of the expedition and from whom we've all learned so much, because he is the expert in growing these extreme microbes. And finally, the last reason why we want to study hydrothermal vents is when we're looking for life on other planets, because it's such extreme environments. So here we have these weird graph. I'm going to explain it to you, but here we have the limits of life that we have observed on Earth, 0 to 120 degrees Celsius, the salinity, the pressure, and then it's compared to other planets and the range that they have. So this is life and this is the Earth. So we can compare the temperature of the Earth to the temperature of other planets and so forth. And we can see that there actually are other planets that are having very similar conditions where life could potentially be. So IODPP is working with NASA to study exobiology, or potential life on other planets. We're talking a lot about Mars right now with Perseverance that is grabbing samples. So this is work that can be related to what we're doing with Mars right now. So we collected our samples. So this may look like a complicated figure, but we have all the cores. And each of these rows, when it has an R to a number, is one core. So we started with long cores, 9 meters, but it wasn't working really well. So we shortened every time we would have a core on deck, would be 4.5 meters. Most of the time, we would not get 4.5 meters. It's hard rock. The drill is breaking the rock, so we would maybe be getting 1 meter if we would be lucky. Out of the 400, out of the 4.5 that we drilled, we would maybe get 1. And in green or yellow or red, it's when we have microbiology samples, and in black, it's when we didn't have microbiology samples. And you can see here, you don't need to, you can see that there's different colors. That means it was different kind of rock. So we got microbiology samples that are representing the different geologies that were present at Brothers Volcano, whether it's at the cone site or in the caldera. And here with all the names of the cores, it doesn't really matter, but they all say R because they were drilled with a rotary barrel drilling bit. This is what our samples look like. This is just an example, but we would basically get a whole round and get this whole piece would go for microbiology. And those are hard rock sample. We had to remove the outside to prevent contamination, because when you're thinking we have to get DNA or try to find cells in hard rocks. So there's probably more bacteria in the air, on my hands, definitely more than in our samples. So we have to be extremely cautious as to, and the drilling fluid, it's seawater that they're using. So we don't want the seawater to contaminate our rock. So we would remove the outside and then separate our core for different measurements, whether it's activity, like viral production. We would split them among the different microbiologists. And I would have liked to present to you some of the results from this, but with COVID, it got very slowed down, and we actually got in my lab our first results last week, so I didn't have time to analyze them. But at this point, you're probably wondering who inhabits Brothers Volcano. I cannot tell you who inhabits Brothers Volcano below the seafloor, which was my part. But in December 2020, sorry, they actually looked at who is living in the chimneys. So where the fluids are coming out, you have these chimneys, and microbes love these places because as soon as you're outside, like on the outside of the chimney, the temperature is livable and it's full of nutrients. And what they found is that there was a mix of bacteria and archaea. There are some sulfur oxidizers. So I told you bacteria can breathe other things than oxygen. So in this case, they would breathe hydrogen sulfide, which we cannot do. There are also some that can use iron. And and you can see between the cone and the caldera, the group of bacteria, each line here is a different group of bacteria. They are different, but they are different because here we have very high temperatures. This is where the hot fluids are coming out. There it's more diffuse vents, so the temperature is not as high and there's more seawater that is mixed in. And because maybe not all of you are interested in microbes, you may be wondering who is also living at Brothers Volcano. Did we see animals? We could actually see often shrimps or, and here I'm gonna show you, it's not going to show just because, wait, I'm gonna show you the video. I want to show you this video of a shark. I will get, oh, there it is. All right. So this was the first caption of a ghost shark on camera. So we can see the drill bit. We were trying to find we can see the seafloor, and there is this little ghost shark that was swimming around the drill bit. So that was the first time that this type of shark was actually caught on camera. He's very cute. And without further ado, if you guys have any questions, I will be happy to answer. And so I showed you a picture of the scientist party, but this is everybody that was on board, from the drilling crew to the people who would like cook for us, clean the rooms, like the technicians, because scientists need help because it's all equipment we're not used to. So this is the whole the whole party that was on board. So half the time, one half would be sleeping, the other half would be working.
Well, after 2 months together at sea, I guess you made some friends.
Yes, I did. Yeah, and I actually, Viviane Pellizzari was another microbiologist, and she was from Brazil, and she invited me to give a lecture at the Brazilian Society last year. So I got to go see her and talk about my science at the same time.
Fantastic. Seeing that video and the drill bit, you know, I'm wondering how are you targeting? Do you know anything about that? How they target that drill?
So they often they compare it. So it's like you would be on top of a ladder with a long string and a needle, and try to go in a very specific hole to put your needle. These guys do magic, but usually what happens, they will go and drill a hole and put a cone. So the cone has like black and white, so they can orient the ship, but it's still very hard because the drill bit is 1.5 kilometers long and dangling from that tiny boat, and you have the waves that you have to be careful to.
Yeah, that's amazing. That's like refueling a plane. Amazing. And when you were talking about where you would sample on the volcano, you know, top of cone, side of cone, tell me a little bit more about that, just to, because obviously you would get deeper or not, you know, based on the ability of the drill.
Yeah, so actually we had two amazing holes, one that was on the caldera, and we were expecting that because it's not as hot, it's on the outside, the outskirt. But when in the cone, we were not sure how deep we would get, and we got to I can tell you exactly how deep we got. We got to like almost 350 meters below the seafloor in the upper cone. So we were directly in the middle of the volcano, drilling 350 meters below the seafloor. And those sites were chosen to answer the objective, but to go from hot to not so hot. The cone is more recent than the caldera. The caldera is the collapse, so we were expecting different geology as well.
Okay. Doug's looking to know, is there a temperature sensor on that drill? How do you know the temperature as you're getting deeper and deeper? Do you know the temperature?
We didn't know the temperature. So the way it works, they will drill, get the cores. And obviously when the cores come back, because they went through the seawater, they would not be hot anymore. But once we're done with the hole, that's when they send the drill, and they have multiple sondes that they would send down. Some that would take pictures of the whole core, some that will measure temperature. We had some that would grab samples, but at least one sort of melted in the hole because it was so hot and corrosive.
Yeah.
So yeah, it's afterwards, and when the drill actually broke, that was when they were doing all these measurements, because for a while you can push seawater, which is cold, but then after a while you you want the real temperature, so they stopped pushing cold water, and that's when everything went south with the drill bit.
So limited by our technology. Jane would like to know, tell a little bit more about keeping that core sample pure for the scientists.
So we did have this clean bench that was actually like moving air away from our samples. we would be wearing gloves, masks, like both like for our eyes, because we were breaking the rocks with actual a chisel and a hammer, so we didn't want anything in our eyes, but also mouth masks so we would not breathe on them. And as remove the outside, anything that has touched the drill fluids. And then afterwards, we would take the samples, immediately freeze them at minus 80 degrees Celsius. So at minus 80, nothing moves. We also preserve some of the samples in liquids that actually stops any biological reactions. So we can work with DNA and RNA, which is unstable.
Thank you. Lynn's looking to know, when your drill bit broke, what happens then? Is there a substitute drill bit, or what happens?
Because they never know how deep they're going to go. They always have extra material on board, so we were able to continue. We had to leave that hole because the drill bit was contaminating everything in there. But there was enough to actually have another, like go to the next hole and just continue making.
Move on. Okay, great. Oh, here's a animal question. Susie would like to know, could you tell approximately what size that ghost shark was?
I believe it's like about, like, it's no more than a meter long, but I'm unsure because that's estimated from the video as well.
Right, based probably on the size of the drill you were sending.
About this big at the end. So yeah, I think, and we saw it multiple times, the shark. It was always there because it's probably feeding off of the shrimps that are feeding off of the nutrients that are coming off of the seafloor.
Oh, wow. Very good. Okay, comment or two about absolutely fascinating. Thank you. That is amazing work that we get to see pictorially here through Zoom. It's amazing. Thank you for your work. Thank you for helping us understand a little bit more of what happens in the large ocean, and a little bit more of what the origins of life might be. One of the biggest questions that we all seek to find an answer. So Dr. Dr. Jessica Labonte, thank you so much. We hope you at some time get to visit Virginia. The southeast corner is pretty beautiful. So when you happen to be in town, please come to the Virginia Living Museum and tour our museum with us. And just this evening is, it's thrilling, honestly, for me as someone who has always loved science, not from the scientific field, but from the business side, and studying and just getting a little bit of your knowledge about these, potential answers to evolution is amazing. And to understand a little bit of the extremities that we have to realize could be a part of that. Particularly with our next series speaker. So we do this, Jessica, monthly. Next month, you can join us via Zoom if you like. We're going to be charmed, I'm sure, by Munaza Alam. Apologize if I don't have that name correct. Munaza Alam is a PhD candidate at Harvard Department of Astronomy. He is a current Nat Geo Young Explorer and National Science Foundation grad research fellow. So he'll be talking about weather on other worlds and studying those atmospheres of some of the exoplanets that you happen to mention. Again, the extremes. So we will learn more about what we might be able to understand from those exoplanets and the extremities. I want to thank, of course, our sponsor, Virginia Health Services, also invested in our community and adult education. So Dr. Labonte, thank you again. And thank you all
very
who
much.
joined us. Have a great evening.
You too. Thank you.
All righty. Bye-bye.
Bye.